We use the simple annotated tools and the variant analysis and for validation RT-PCR and Sanger sequencing and the IPA for some of the genes pathway and which genes have been mutated we could trace. This is the methodology followed. And sample size I know is less to start with a very systematic analysis. We started these 4 cases. There are 4 cases of year of diagnosing, as there were 10, 18 and 2016, and the age is from 47 to 62, and also the 2 males and 2 females which are available with fit into their exclusion criteria, and then when the clinicians are supported that this is the cases we can take, and also there are the clinical tests they might get MRI, CT scan and M-spike T and bone lesion. When you start analyzing the Sanger Institute data of 35 cases, we just summarize, they identify 52 driver mutations, and most of the cancer genes like KRAS, NRAS, BRAF and TP53, they were the most prominent cases they found. This was found in all the 35 cases there, and we identified there also the one which is not reported in the literature is 3 genes, ROBO1, LTB, and these were identified in those exome data. And also we found in the driver gene different kinds of the variations we found. 54 missense variations, 2 splice donors, 2 splice acceptor, and 2 in splice region, and 2 stop loss and 8 stop gain variation we found in these cases. When I compile all the 4 cases exome data, our own data, see the major one which we could able to see with different kinds of filters we used to analyze this data, exome data. Only I am presenting the one which has the major considerations here. Like in case 1, we had 14 different kinds of genes have been involved. In case 2, 13 genes, and case 3, 11 genes, and case 4, 10 genes. There are common genes in these 4 cases. We have taken care to select the samples to have more of homogeneity by inclusion-exclusion criteria. There are different colors indicated here. There are genes, common genes in all the 4 cases, but we do see a different genes is defective in different cases. So even though clinically we made the homogeneity, there are genes involved in this multiple myeloma is heterogeneous. That means different genes have been contributed to cause the multiple myeloma, the case 1 to case 4 there. Different mutations we could see, and also vary in the position of mutations. We analyze in greater detail about with the gene and the variations and what kind of variations as already quoted there. To summarize the variation, what we analyze the structural variation 4 cases, there are insertions about 4,000 insertion case 1 and 59 insertions in the case 2, and 49,000 for case 3 and 35,000 of case 4, and deletions were more in the case 3, 1,600